कुलकर्णी सर.
झूमला अनम्यूट कसं करायचं?
हॅलो अहिरे सर.
येस सर. Shall we start?
पुनवणे सर आहेत जॉईन झालेले.
Okay.
पोस्ट केले त्यांना.
Yeah. So shall we start?
We'll start in 5 minutes, sir.
Okay. Hello, Dr. Parshuram.
Hello, hi.
Okay, we'll start with the session.
Yeah.
Okay, so good morning, friends, dear students. Today we are here to have a national level webinar, one-day national level webinar on research and innovation in life sciences under Rashtriya Uchchatar Shiksha Abhiyan. Honorable Principal Dr. B.T. Jadhav sir, Vice Principal Dr. B.Y. Deshpande sir, Dr. S.H. Mujawar, Dean Research and Development, and Mr. Sushant Pore sir, Chairman IIC, my colleague Dr. Pankaj, and all the faculty members and dear students. The motto behind this webinar, this one-day webinar on research and innovation in life sciences, is to induce the students of life sciences particularly. And we had organized a series of webinars on these topics for different branches of our institute, like chemical sciences, physical sciences, mathematical sciences. And today we are here with the webinar on for the life sciences students. So for this particular webinar, we had invited two eminent personalities with us, and we had two sessions for today's presentation. The first session will be dealt with by the Dr. Parshuram Sonone, and the second session will be dealt with by the Dr. Sayantan Panda from Israel. Okay, so the first for the first session we had with us Dr. Parshuram Sonone. He has completed his MSc from Department of Microbiology, University of Pune, now Savitribai Phule Pune University, Pune. Then he did his PhD. He qualified the NET, SET and GATE examination when he was appearing for the MSc. And then he joined his PhD at Indian Institute of Technology, Madras, under the guidance of Professor Nitish Mahapatra. After completion of his PhD, he joined a postdoc as a postdoc fellow at Indian Institute of Technology in 2013. Then from 2014 to 2018, he had joined as the postdoctorate fellow at National Center for Cell Sciences, NCCS, Pune, under the mentorship of Dr. Radha Chauhan. Then he also had the NPDF work as an NPDF fellow from April 2018 to January, and then DST NPDF fellow from January to last month. And last month only he had joined the Rajiv Gandhi Center for Biotechnology, Bharati Vidyapit Pune at Dhankaudi, Pune. He has around 13 papers to his credit, and he has attended several research conferences to present his work. He has been awarded as Junior Research Fellow, then CSIR NET. I already told you that he qualified the GATE, then NPDF from ACRB, that DST, and NPD, the RA from the DBT. So he is very keen person. He is basically a microbiologist. And I request Dr. Parshuram Sonone to deal with his presentation.
Thank you, Mahendra, for your kind introduction. Am I clearly audible?
Yes, yes, sir. Yes, sir.
So
now you can share the screen
start
as you are the
video
host.
as well. Yeah, I will do that. Before presenting, I would like to thank Yashwantrao Chauhan Institute of Sciences and Principal Professor Jadhav, organization committee for this particular conference. Mahendra, especially you for inviting for this talk. Thank you very much, all of you, for attending this particular seminar. So today I am going to present my work which I have done in NCCS. Although I have done a lot of work in IIT when I was a PhD student, since it's like we have limitation of time, I think I will focus only on that. Okay, so I will start my presentation. Yeah, is it visible? My presentation is visible?
Yes, sir. Yes, sir.
Yeah, we are on the first slide, right?
Yeah.
And just a minute, is it changing now?
Yeah.
Okay, so I can start now. Okay, thank you. So in NCCS, we work on something called as nuclear pore complexes, studies on central transport channel of nuclear, excuse me. Studies on central transport channel and Nuc 93 complexes. I will come to this definitions in detail in upcoming slides. To start with, I will start with introduction to nuclear pore complexes. I think most of you know what are the nuclear pore complexes. Still, I will explain. So every eukaryotic cell has different membrane compartments, and nucleus is one such compartment which stores the genetic information of the cell. And this particular information which is present in a genetic material needs to be communicated with cytoplasm as well as the external signals from the cells. So to communicate this information between the genetic material and the outer environment, nuclear pore complexes act as a gate. These are multi-protein assemblies present in the double membrane of nuclear membrane. So nuclear pore complexes are mostly made up of proteins. There are 30 different types of proteins which make nuclear pore complex. And there are multiple copies of each nucleoporins. So the nucleopore complex proteins are called as nucleoporins. So there are 30 different types of such nucleoporins, and each nucleoporin will be present in multiple copy to assemble into one functional subunit of nuclear pore complex. So in eukaryotic cells from cell type 2 organisms, there are differences in number of NPC molecules present in the nuclear membrane. They range from 10 nuclear membranes, 10 NPC copies to even 4,000 to 5,000 nuclear pore complexes in one single nucleus. So in general, we can subdivide this nuclear pore complex into different types of parts based on their organization and their composition. The most commonly known as cytoplasmic filaments, which are present towards the cytoplasmic side, and the nuclear basket, which is like a basket-like shape, which is present towards the nucleoplasmic side. Apart from that, there is an important core and membrane interaction region, which actually adhere this entire assembly to the nuclear membranes. And at the core of NPC, there are two important complexes known as coat nucleoporine complexes as well as adapter nucleoporous complexes. So these particular complexes are assembles of different proteins. We will go in the next slides to see which type of proteins are making this particular different subunits. So the most important function of nucleopore complexes is to transport various types of molecules across the cytoplasm and nucleoplasm. So if you see in a general, I think you guys have studied in probably in zoology where you see the different mechanisms to transport cargo inside the cell. And you have heard of this RAN cycle, RAN GTP gradient cycle, which is used as energy to translocate components from cytoplasm or nucleoplasm to each and vice versa. So there are two types of nucleocytoplasmic transport. One is active and another one is a passive. The passive one are like small molecule which can easily diffuse through the nuclear membrane, and they don't need any energy. But in case of larger molecules, such as the proteins of signaling pathways, the proteins which are essential for the cell functioning, they need to be translocated through the energy-driven pathway, which is generally known as RAN GTP cycle, RAN GTP, RAN GDP. The GTP is hydrolyzed to use or generate the energy to perform this particular function. So the input of various molecules such as viruses as well also needs the mediation by this RAN GTP pathways. Apart from that, there are several processes which are happening in the nucleus. They need to be like facilitated through NPC, such as, you know, that ribosomal proteins, although they are synthesized in endoplasmic reticulum, but they need to get assembled as a ribosome inside the nucleus. And those assembled ribosome particles, which are larger in the size, they also are needed to be diverted to the cytoplasm through nuclear pore complexes. And this is one of the gated mechanism. So there are nucleoporins which have something called as repeats of phenylalanine glycine. They have anchoring sites for these cargo molecules. They interact through it and then they pass the molecules outside the nucleus. So that's why nuclear pore complex play a very vital and important role in the cell physiology. So despite of knowing that, we know like there are like different types of proteins which are like making nuclear pore complexes. So since as I said that there are 30 different types of protein and different components are made up of different types of nucleoporins. So for example, here I'm showing in the green highlight that the nucleoporins, which are transmembrane nucleoporins, NDC1, GP120 or POM121, these are the nucleoporins present in the nuclear membrane. And they provide as anchor to the other complexes, such as the adapter complex, which is made up of 5 different proteins, such as NUC155, NUC205, NUC93, NUC188, and NUC35. So generally in nuclear pore complexes, the molecular weight of the protein is generally assigned as a name to the protein. NUC155 generally refers nucleoporin, which is having approximately 155 size. And the another important component which I studied was the central transport channel, which is made up of 3 nucleoporin, NUC62, NUC54, and 58. And this particular protein is present at the mid plane of nuclear pore complex and is very crucial for the controlling the path of molecules across the NPC. So this is the number of proteins or the names of protein which I'm showing for the human nuclear pore complexes. Apart from that, there are differences in the names as well as composition in NPCs of yeast as well as the other organisms. Until 2010s, people used to believe that the nuclear pore complexes are kind of conserved. That means if you take the human nuclear pore complexes, the complexes would be similar if you go from human to even the yeast as well as the even lower organisms. But the recent studies show otherwise. So when we started working in NCCS with our lab, we had this big challenge because we knew that our hypothesis was that yes, indeed, there are lots of differences between the nuclear pore complexes of lower organisms to the higher organisms. But we had a lot of counter arguments for this, that no, it is conserved and so on. But later on, I think our studies have established this fact that, and now in the field, I think if you see 2020s, people are now talking about these differences. So one of such review which we recently have communicated shows or summarizes these differences. And I can tell you that these are the cryotomographic structures of nuclear pore complexes. So this is one of the new method which has developed in recent past, probably last 2 decades, where you can actually analyze nuclear pore complexes or any larger cellular assemblies by utilizing electron tomography. It's a combination of the electron microscopy with tomography at the lower temperatures. And people use a lot of computational method to get this particular information. And if you can see that these are the different tomography maps of nuclear pore complexes obtained from different organisms, such as Saccharomyces yeast, Chlamydomonas, which is a unicellular organism, Dictyostelium, I think you know Dictyostelium as well as the Xenopus, and the human one. So if you can see that, just see these maps, 3D maps, and you can see that there are prominent visible differences, which something like if you see extreme right, which is human one, where you can see that the length and bulkiness of nuclear pore complexes of human is higher compared to the yeast Saccharomyces one, which you can see in the extreme left panel. If you see the cross-sectional view of these nuclear pore complexes also, you can see that there are still fine differences between the organization of these particular subcomplexes. So although these structures are available, the resolution of these structures is really not really high. The best is still 22 angstrom. And with this resolution, we will not be able to differentiate the molecular organizations of these particular NPCs from 2 different organisms. And this is something a challenge in the current nuclear pore complex field, to obtain the structural details of this nuclear pore complex. So when Radha Chauhan joined lab in NCCS in 2014, so that's the aim she started with, whether we can get the high resolution structures of nuclear pore complexes. As I told you that it's a big challenge for structural biologists as well to get the structure of nuclear pore complex, because it's already made up of 30 different types of nucleophores, and there are at least 2,000 copies of different proteins in one single assembly, and that makes it is really challenging. So this particular challenge is also compounded by the presence of structural diversity present in the subcomplex level as well. So in this particular case, I am showing the 3 nuclear pore complexes from human, yeast, as well as Chlamydomonas. And you can see that the top ring and nuclear ring, which are present in the nuclear pore complex, you can see that these are the cross-sectional views of these 3 rings from human, Saccharomyces, and yeast. And you can visibly see that these complexes are having a really distinct organization. And this particular organizational differences are nowadays people are trying to people started to talk about. And these particular complexes are made up of multiple copies of something called as Y complex. So these Y complexes are arranged in head to tail fashion, in the circular fashion. And you can see that in a middle figure where you have only single Y complexes, you can see that they are arranged in 1 to head to tail organization. And this structural details of the proteins from human as well as the yeast of individual components now play important role in understanding these complexes at the large level. So this is something, one of the studied complex, although we don't have high resolution map of pi complexes from like these two species as well, but still the information available from the individual protein actually is like very essential to understand the differences and the organizational differences in this particular NPCs. So the understanding of differences of organization is very essential to explain the function of nuclear pore complexes, which I will come to the later part of the presentation. And that's why it is very crucial to get the structural details of nuclear pore complex subcomponents or subcomplexes. Apart from that, so since we started working on human nuclear pore complexes, we had a big challenge because there is ease with yeast nucleoporins. They are very easy to handle with. They are easily solubilizable. And mammalian nucleoporins is something, what is the challenging part, that it's very difficult to purify mammalian nucleoporins. And apart from that, it's like needs a lot of detergent to purify when we are purifying mammalian nucleoporins. I think similar problem is there with the plant nucleoporins as well. And because of that, it becomes very difficult to crystallize the nuclear pore complex. And people have started using different organisms, but still now, as I presented before, that utilizing the lower organism provides good insights on nuclear pore complexes. But then when you want to study human nuclear pore complex per se, you need to get the nuclear pore complexes or the nuclear pore from the human sources. And this was the challenge which we had in Radhachand's lab when I started as a protein biochemist in this lab. So what particular approach we wanted to use was the integrated type of approach, where instead of focusing on the entire nuclear pore complex, we'll take the subcomplex, so small subcomplex of nuclear pore complex, and start studying that to get the atomic detail. So I started working on this central transport channel, nuclear CTC and NUC93 complex. As I think I remember, I showed you this figure, and you remember this is present on the central plane of nuclear pore complex. Now this particular region is shown in the amplified version here, where you can see that NUC53, NUC62, NUC58 interact with each other. And this particular complex interacts with another subcomplex as known as the adapter complex. The main constituent of adapter complex is a protein called as NUC93. Its molecular weight is 93 kDa. This particular 93 interacts with another set of nucleoporins in the NUC93 subcomplex. Altogether, these two complexes make the major chunk of nuclear pore complex present in the central plane of NPC. And so do that, what we did, like we utilized the combination of electron microscopy, IP, as well as immunofluorescence based studies to get this particular complex. Something which I am not showing in this presentation is that when I started, actually I started purifying only NUC93 protein. And to my surprise, after spending 1.5 years, that we realized that we've tried almost all possible methods of purification, expressed in human expression system, mammalian expression system, bacterial expression system, utilize different methods, different tags, and still this was not solubilized. But then I think eventually, since I had different backgrounds, so since I was trained in IIT with the different methods, so I started with my strongholds, like whatever methods which I was like good at, I started with that. So to start with, where we started is like we started at NUC93. We focused on first NUC93 because this particular protein interacts with at least 7 different nucleoporins. And then we decided, okay, we'll focus on NUC93 and then chop out like what are the essential regions of NUC93 to interact with different proteins. To start with, we started with CTC. So what we did, we cloned full length of NUC93 from human eukaryotic cells. We cloned that into a vector called as EGFP-C1. So with that, what happens that you get GFP tag to NUC93. Based on secondary structure predictions using different software such as Cypred, we identified what could be the potential secondary structural element such as alpha helices, beta sheets in this particular structure. And based on that, we made several deletion constructs of that, and we cloned them as well into the GFPC1 construct. So we got different Nuc93 constructs tagged with the GFP. So with this particular set of constructs, we did something called as immunoprecipitation. So in case of immunoprecipitation, what we do is generally use the antibody against the protein of interest, pull it, and detect the interacting partner, say for example, protein X, which interacts with the protein of your interest, which is tagged. Pull down with antibody of protein A, and then detect with the protein B. So in this particular case, what I did is that I transfected this particular set of constructs to human HEK293 cells. And after getting the expression and lyse the cells after 3 days of expression. After lysis, we pull down with anti-GFP antibody. Or in this particular case, we use specifically called as nanobody. So it's a version of antibody. And with that, we pull down Nuc93. So as we know that 93 interacts with CTC, when we check the presence of CTC with the full length Nuc93 in this second panel, you can see that we can see the signal for central transport channel. This particular antibody, MAB414, recognizes the FG repeats of Nuc62. And that was the good start for us, where we started that yes, Nuc93 can interact with CTC. Now, since we had several deletion constructs of this, and we could find that Nuc1 to 150 region, which was high, like almost 1/10 of this entire protein, this region itself was sufficient to interact with CTC. And that gave us good confidence that only this particular domain was essential for the interaction with CTC. And then we made several deletions of Nuc93 as well, where we split this entire region into 2 parts. And to our surprise, what we saw that 1 to 82 region itself was sufficient for CTC interaction as well. So our studies, we focused on these 2 constructs, where one was 1 to 82 as well as 1 to 150. So to confirm the functionality of this particular region, whether this particular region is like not just artifact of interactions, but it had some functional role as well. So we utilized immunofluorescence methods, where we perform the confocal experiments with the similar constructs which were GFP tagged. And with that, what we could see that 1 to 150 region, you can see that it was able to localize to the nuclear ring. And this is the cross-sectional view of each nucleus. And you can see that these maps, which you show that the GFP signal or Nuc93 signal actually corresponds to the nuclear ring, which is present in the red. So since full-length protein, as we can see, is expected to localize into nuclear ring, and we could see the strong signal. And you can see in the smaller panels here, we can see that round circular rings of nucleus present in the detected with the Nuc93. Interestingly, we could see that 1 to 150 region also was able to make such kind of the rings. That means that this 1 to 150 region was able to get into the nuclear pore complexes. And to our surprise, the 1 to 82 region, which interacts with CTC, actually was not able to get into this particular ring. And that had some explanation, which we will talk in the next slide, where we could see that this particular 1 to 150 region entire region is essential for interaction with NPCs. And something which I was having a problem so far, where I was not able to purify Nuc93. So now next thing what we did, that we took this 1 to 150 region and we co-expressed all 4 proteins like Nuc62, Nuc58, Nuc54 and Nuc93 in E. coli cell, and we could actually purify using histag first, and then followed by GST, and then followed by size exclusion chromatography. And you can see that this is the last step of the size exclusion chromatography, where you can see that single peak harbors the 4 proteins of this particular complex. And this was like big thing for us, like big purification of single protein itself is a challenge, that we were able to get a stable complex. And this particular result actually encouraged us to look for the full length protein as well. And to our surprise as well, when we clone this full length protein in GST tag vector and co-express with these three CTC nucleophilics, and we could get this complex, and we could actually make this 4-protein complex. And this particular complex is really important in the stage like for the structural studies, as we this is something which is like one of the central region of transport channel of nuclear pore complex, and this information will be really useful. So what we did next step, that we took these purified complexes with full length of 93 and did something called as negative stain electron microscopy. So this is something, I think, a new field where people have started using electron microscopy method to determine the structure, which is like better, which has some advantage over X-ray crystallographic methods. Although these methods are also have challenges, something like what we have faced. And you can see that in electron microscope, under electron microscope, we could see small particles. What we do in this lab, in our lab, we take this different micrographs from the sample and use computer algorithms to understand the structure. So what we do, we take the snapshot of each particle, and we do something called as particle averaging. So after that, we can classify different orientations of these particles from these 2D images. We call as 2D classes. And you can see that you have this noisy image in this particular picture, but when we do average thousands of such particles, we can start seeing the structural features. And this is particular thing what I'm showing here in this panel B here, where we have different orientations of one three-dimensional object. And in this case, this was our CTC and 93 complex. And using the different algorithms, we could get three-dimensional structure of initial three-dimensional structure of this particular complex, where we could get the information about the organizational details of CTC and NUC93. So the red molecule here is NUC93, and then 3 molecules of CTC are shown in the 3 different colors. And this particular information gave us the clues that this particular organization of molecules is in V shape. And probably we need to add probably some more of molecules to get higher details. So we have, because of the COVID, I think we could not get a lot of time for this electron microscopy. But to get higher resolution structure, we need to do cryo-electron microscopy, where instead of using negative stain, we have to use unstained sample and perform the similar analysis. And this is something which lab is currently doing in NCCS. But I think we published this particular what we could show that these nuclear pore complexes are not the static. So if I think so far, whenever I was presenting the image, what was printed in your mind was this particular complex is kind of static. Static means it is like rigid, but no, that's not the case. The people over time have proved that the NPCs indeed are dynamic. That means the number of proteins which are present in the NPC, they constantly get exchanged with the time. And what we could see that the observation which people saw with the entire NPC with the live cells, we could actually demonstrate them with in vitro as well, where we could see that the complexes of the CTC actually were very dynamic. So in this particular case, we use something called as size exclusion chromatography coupled with multi-angle light scattering. This particular experiment will help us to tell whether there are like dynamic complexes present in the solution or not. Although these complexes are made up of only 3 different proteins, we could see that by sigma analysis, they are having different type of molecular weights. Different molecular weight complexes are present in the solution. And that gave us the clue that yes, indeed, these CTC93 subcomplexes probably are dynamic. And overall, this particular study established this fact that dynamicity is present not only in entire NPC, but in the component or subcomplex of NPC, as well as having encoded the information for this particular dynamics. And so this particular study, with this study, we could see that 1 to 150 region or N-terminal domain was essential for the interaction of NPCs. And what we could do, like we could purify NUC93 from human sources as well. And that was the thing which we showed. There are like a couple of manuscripts present in bioarchive from our group, which show that this 1 to 150 region, which I talked about, has 1 to 82 region, which interacts with CTC, but the region from 96 to 150 interacts with the other nuclear pore complexes, such as nuclear pore complex proteins, such as NUC205 and NUC188. So together, the entire region is essential for anchoring several proteins of adapter complex of nuclear pore complex. And this is what we could show. Apart from that, we could show that there's dynamic nature of complexes across the various species of NPCs. And this particular complex, which we could reconstitute in vitro conditions, and now serves as the basis for making higher complexes of human subcomplexes. And currently in the lab, some people are working, PhD students are working, and they are having success with making 5 protein as well as 6 protein complex. And soon probably we will get the information for that as well. So with this, I would like to thank different people associated with this study. Dr. Radha Chauhan, who is the guide for this work, and Dr. Janesh Kumar, as well as the directors of NCCS and the people from IIT Delhi, where we did this negative stain electron microscopy, and then from University of Hyderabad, where we did final data collection for this electron, the fluorescence microscopy, and then several people of Laboratory of Structural Biology and CCS. So this is what I would say I have done for this particular time interval. So I'm okay with the questions. And then after that, I have one small part, because I think, as Mahendra told, that there are a lot of students interacting with it in this session. So we'd like to give guide like some idea how research is pursued in the big institute. But I think I would like to take questions on this particular part of presentation from all. And after that, I think I will deal with the small portion for the students. Yes, you can ask the questions.
Students, if any questions are there, then they can raise their hand or put it in the chat box. Okay, sir, can we move forward so that student will ask if
yeah,
mostly
maybe they can type the question
have
in the chat box and then read those questions for me, please.
Okay, no problem.
Yeah.
So
okay,
we'll move forward.
Yeah. So with aligning with this, the objective of this particular theme. So although I have very little information about this to tell here, but what I can do is like I can integrate my personal experiences with the students here, and then and that will be good for the student, I think. So if you see in India, we do different type of research. So we can classify them as a basic or fundamental research. Then there is like applied research and developmental research. So in a basic or fundamental research, we generally pursue the certain types of phenomenon or certain type of hypothesis, which is very crucial for explaining the natural mechanisms in a natural sciences. And this is very important step, because that particular information is very crucial. And today, as we can see in this COVID world, we can see that although the COVID was a new kind of virus, but within small span of time, we could decipher several things. And then this credit goes to the lots of fundamental research people are doing from last 5 or 6 decades in life sciences. Another type of research is applied research, where we know that certain phenomenon, how they work. And you can make utilization of these phenomena to apply to the community as well as at the industrial scale. And this is also very important part of research, because of that we get to know so many things. For example, I will tell you the example of penicillin discovery, and you can say that the discovery of Alexander Fleming is classical, basic, or fundamental research. But what Cohen and Chain did, that the purification of penicillin and utilization it to the utilization of it for the research, the therapeutic purposes, is kind of combination of fundamental as well as applied research, and this is something very essential. And then there is also another kind of research people do, is called as developmental research, where people try to develop different types of streamlines or mechanisms to study as well as get the higher yields of some products also. So if you see the scenario of funding in India, so still this is the national R&D statistics data, which is for all different fields. This is not just life sciences. Most of the research still in India is funded by the central government and the private sectors. Although this particular data set shows that it shows 38% of the contribution from private sector, it mostly comes from the automobile sector as well as the software sector. Very little from the life sciences or pharmaceutical sector comes for the R&D in the life sciences. The contribution is still less, and most of the funding for research in life sciences still comes from the central government. So for a student, if they want to do research, they need to get into some research institute, and there are several bodies of research. They are found by Government of India. So like this Indian Council of Agriculture Research, which promotes the different aspects of agriculture sciences. Similarly, there is the Indian Council of Medical Research, which is dedicated towards the medical and pharmaceutical type of research. Apart from that, there are independent government ministries such as Department of Biotechnology or the CSIR, Council for Scientific and Industrial Research. These are the different bodies which provide the funds for doing research. So all these institutes, even like UGCs as well as the ministry, MHRD, so they also have a lot of institutions such as IITs, ICERs, NITs, NIPERs, or so on and so forth. Apart from that, there are independent research institutes such as IISc, Indian Institute of Science, which is located in Bangalore, or the institute like NCBS present in Bangalore as well. Apart from that, each of these funding bodies, they have their own institutions as well. So for example, NCCS, which I studied in, was one of the biotechnology institute, and biotechnology has at least 16 such institutions in different parts of the country. Similarly, CSR has like lots of laboratories all over the country. And the very well-known example from the Pune is National Chemical Laboratory, which is present in the Pune, and which does different type of research. So similarly, the ICAR also has lots of research institute, such as many agricultural research institutes present in India. And then their famous institute is also like NDRI, National Dairy Research Institute, where lots of research goes on the dairy and dairy related product. And ICMR, I think you guys know very well with this COVID, and you know that the Dr. Bhargav, the director of ICMR, was key person in developing the Covaxin, the indigenous vaccine for COVID-19. So this is how the main infrastructure is generated in terms of funding by Government of India. So these institutes have different laboratories. There will be like n number of scientists, ranging from 10 number of scientists to up to 350 scientists present in one institute. Each scientist has different domain of their own research. And now as a student, what you need to get is like, you need to first get into this institute and then get into the different laboratories to do the research. And what is the key or what is the first step to get into this institute is to qualify some of the these fellowship programs, such as the JRF or SRF fellowships from CSR, as well as ICMR, ICAR, DPT, and then even like this, like a couple of years back, Prime Minister started new research fellowships as well. Apart from that, I think there are like SERP also provides a lot of fellowships to the people who just have completed a master's and want to get into research. So these fellowships come with the funding as well, because they provide some amount of fellowship to support the student. There are some institutes and where people, especially the people from the rural, that's what they have hardships with, that they are not able to qualify these exams, but they are actually are brilliant. So there are some exams like such as GATE as well as JJ where people will, once they qualify these exams, the research institutes which are hosting these students pay them, or they support the fellowship. So something like GATE, where IITs will provide the fellowship to the student who have qualified the GATE. And this is how it particular scenario is for the students. So this is the basic scenario for the getting into the PhD programs. I think there are two more steps which I think nobody talks about, but I think I would like to talk about that today. So once you qualify these such exams, either JG, GATE or CSIR NET, you need to apply for several institutes depending on the interest and the background you are having. So make sure one thing, that the time is changing. So people initially used to have this concept: if I have done MS in microbiology, I should only pursue the research in microbiology. It's not the case actually. So most of the labs actually are having interdisciplinary research where people study from different subjects. So be open for this and find out what would be your interest in the research. That's one thing. Second important part is the interviews, where people will be asked the interviews what they have done in their masters. And most important part in these interviews is generally the thesis work they have done, or the project work what they have done in their masters. And then very important criteria to evaluate the student is how much you really understand that particular part. And what we have seen, as at least from last 5 or 6 years, I'm closely observing the students, and most of the students actually fail in this particular aspect, where they will not be able to explain, because if you are not able to understand it, you will not able to explain. So I think people need to pay a lot of focus on that, where they read about the project very well. Even they have done one single experiment, that's okay, but the experiment should be able to explain like how they are able, how that particular experiment explains certain types of phenomena or certain types of logic. This is something people or the student needs to understand. And then second important part which comes with the many students, even I have seen same thing in IITs as well as many national institutes, that most of the people, even they are able to cross the exams, they are not able to, find out what they really want to do, or what they are, they want to do or pursue in the research. And I think if you really want to get into the research, I think you should be really thorough with this part, where you know exactly what you want to pursue, at least what is your quest. That curiosity of mind should reflect, and this particular thing is very, very essential, and you need to have patience to deal with all this. So I think that explains you the basic infrastructure, or like basic mechanism how to get into this research institute. And I would like to specially tell you guys that don't lose your heart, or don't underestimate yourself. Everyone has a potential. So prepare well and appear for these exams confidently. I think you will get the success. So with this, I think I would like to say all the best to all you guys. And I would like to conclude my talk here.
Now, if any questions are there, then you can ask in the chat box. as the motto of the webinar is to uh, tell students that the peoples who are coming from the rural background can also reach up to the level where the Dr. Parshuram has reached. He is actually our classmate from our BSc standard. So he is also doing the BSc and then MSc, and then he qualified the NET, SET, and GATE, and he joined the IIT, and now he is working as an assistant professor. So in research, there are several opportunities. He had already been explained. So if any clarifications you want to ask, then you can have it in the chat box.
You can ask in Marathi also, that's not a problem.
There are no any questions, then I think the topic was quite heavy to them, but the latter session was really motivate them to come into the stream of the research, as we had lot many opportunities in life sciences research. As
No, I think
particularly,
I personally wanted to present this part because this is what we think that like what we are doing in this like big institutes. So we
yes,
do
that
deal
is
with
that is why we
really
told.
big challenges. Yes, I think even they go back home and then read about what cry was. I think that is like more than enough
Definitely.
for the particular thing. So this is how it is, and that's the advantage of being in National Institute where you can do good research. And then time also is changing, where I think people have started investing in the smaller colleges as well as institutes also. I think this RGITBT, where like the Institute of Bharati Vidyapit also is like one of the good college where people have balanced research as well as the teaching as well, and that helps in students to get exposure also. I think this particular program is organized by the Uchchatar Shiksha Abhiyan, and that's the aim of this particular program, where people or students should get exposure to more and more research. So I think this is a good initiative by your college and institute, and it's a good thing. So keep doing this.
Yes, sir. Yes, sir. Definitely, as we are now moving forward to the cluster university, then again we need to improve our research background also.
Yes.
Whatever we are doing today is quite the basic research, but again we need to look out into what is going in the national institutes and
Correct.
the
Yes.
international institutes.
Yeah, I
So
think this
students
bridge
are also
between,
need to
yes,
look around that what is going on in the world, and then
word
they should decide their problems for their future research.
future research. Yes, and they should get bridged with these institutions. So I think the research is also having globalization now. So you can do any experiment in any other laboratory. You just need to approach the people, and then many of them are like open with ideas.
Okay, so thank you for your
Yeah.
kind your time, as the and your guidance will definitely be fruitful or important for us. And
Okay.
if students later on, if they came to us, then we'll definitely have some move forward to you, keep forward them to you. And if they
Yes.
are required more guidance, then you will definitely guide them. Okay, so
Yeah.
on
Sure.
behalf of the organizing committee, I am very much thankful to you for your valuable time. So students will have just the next session will start within next 5 minutes. The speaker from Israel is being joining within next 5 minutes. So be present online. Don't leave the meeting. Okay. Thank you, sir. Thank you.
Yeah, thank you. Thank you, Mahendra. Thank you.
Hello. Yes, you नहीं दिख रहा है. Yes, you T ना. हाँ हाँ दिखा. नहीं रे, अलग नाम से ज्वाइन हुए हो. MLADY नाम से ज्वाइन हुए हो.
I think he needs to unmute himself.
Okay, so I will start with the next session. I welcome Dr. Sayantan Panda on behalf of the organizing committee. I welcome Dr. Panda on behalf of the Yashwantrao Chavan Institute of Science, Satara, for this webinar on research and innovation in life sciences. I'll just introduce him as he's again one of my best friend. We had spent a lot of time in Pune University when he has did his MSc, MPhil, and PhD. So Dr. Panda has done his BSc from Department of Botany, Bankura, Samilani College, Bankura, West Bengal. Then he joined for MSc at Department of Botany, Savitribai Phule, Pune University, Pune. After completion of his post-graduation, he joined as MPhil student and completed his MPhil and PhD under the guidance of Dr. Avinash Kamble at SPPU Pune. Later on, he had joined as a visiting student at Weizmann Institute of Sciences in Israel under the supervision of Asaf Aroni. He is the very renowned person in the field of plant sciences. He had joined as the postdoctoral fellow from June to 2017 to 2018 as a postdoctoral fellow in the Department of Plant and Environmental Sciences. And he worked on the investing the biosynthesis and regulation of specialized metabolism in plants. Then he again presently working as the a postdoctorate fellow at Department of Vegetables and Field Crops at Agricultural Research Organization, Gillett Research Center, Negev, Israel, under the supervision of Hagai Yazur as a scientist. During his research career, he had published several research papers, which are published in Nature Communication in the top cited journals like Nature Communication, Proceedings of National Academy of Sciences, USA, and all that with high impact factor. He had around 19 research papers to his credit. And now I request Dr. Sayantan to continue with his presentation.
Thank you for the invitation to give me opportunity to talk in this platform. So thanks for the introduction. What I can say about myself that I am just a student. I'm trying to understand what I am doing and if I can use my knowledge for further development of the society and in general in the science. So let's start. Can you see my screen?
Yes, sir.
Hello?
Yes. Yes, you can continue.
So today I would like to talk about how to come to the research and how to do innovation in our life science field and so on. So what include life sciences? So life science is the scientific study of living organisms in their life processes. So how they grow, how they born, how they interact with each other, and so on. There are different streams of life sciences. You might know them like genetics, microbiology, zoology, botany, physiology, biotechnology, astrobiology, and so on. So what is life science and what is biology? Is they similar or they are different? So the life science comprises field of science involving the study of living organisms, plants, animals, humans, bacteria, and so on. While the biology remains the centerpiece of life science, so it's a part of life science. It's mostly deal with the technological advances in molecular biology, biotechnology, and That's how it's build a specialization in new disciplinary fields. Biology is overall natural sciences of studying life, while other life sciences, it's subdiscipline, so it's a part. Some life sciences focus on specific type of organism, like for example, microbiology. Microbiology focused on microorganisms like bacteria and so on. While zoology focus on animal, botany focus on plants, and so on. There are different fields with particular topics. So how do I become a life science scientist? So the basic eligibility for life science. So the basic eligibility, you must know that the candidate must have a master's degree and prior knowledge of basic research. Then second, masters with full research thesis are always an advantage. So here I would like to elaborate. So what is full research thesis? So as I came from India, I understand that where we, like for example, the students from Indian system suffer here. Because in abroad, outside India, in all the developed nations, masters consider a degree with full research. That means there are very light theoretical credit load, and you have full 2 years you should do research in the lab. So that means your research should be something which is novel. You must able to publish something out of it, and that's how you introduce yourself already in research. So further, after the masters, when you go and join for PhD, or you want to go for industry, you join for some company, wherever, you can tell them, look, I already have a research profile where I did work on this. So this give you always an advantage. But for our case, when we do masters from our system, we do thesis, like we don't have basically research thesis. We have a dissertation, which is 3 to 4 month length. And 3 to 4 months, how much research one person can do? It's a big question, right? So now there are also, that's what we do in case of masters without thesis, that we don't have a proper thesis. So after masters, a candidate must have at least 2 years of on-hand research experience. You must know what is the principle of research, what is code of conduct, how to perform research, how to think, and so on. So after masters, a candidate should do at least 2 years of on-hand research experience from somewhere where you have exposure. And after masters, of course, after masters, you can get a job as a scientist. But if you continue for PhD and after PhD you plan to join for a research field, it's always advantage, because PhD give you a kind of a license. Like, you know, if you want to drive a car, you need a driving license. Similarly, if you want to do research, you need a license, and PhD kind of that type of license, that you have a prior knowledge of research already, and you are already a known person. Now you have all this minimum qualification. For example, you have a master's with thesis, or you have a research experience, you did your PhD, and so on. Now you can be a scientist. Of course you can, if you know how to think outside the box. What is outside the box thinking? Outside of box thinking means you should not think how everyone is thinking the similar way, or you should not try to think the question which is already solved, but you think in different way. For example, all of us, we know about Sir Isaac Newton, who, like, law of motion, who discovered. And we heard the stories that the apple fall, so he realized, okay, why apple fall down? So he law of gravity, and so on, he discovered. Now you say apple follow the law of gravity, so you start to understand orange follow law of gravity. No, this doesn't work, because this is not outside the box. You should think complete new stuff which is still not solved. And then eligibility, you should work independently. You should not be asked always, okay, what should I do now? What should I do now, to another person? You should think by yourself what you should do and how you should solve this problem. This is one part. And second part, as nowadays all kind of sciences are kind of interdisciplinary, they are connected. So for example, taxonomy, it could be microbial taxonomy, animal taxonomy, plant taxonomy, and so on. They alone is not stand. It's connected with other field, like for example, biochemistry. It connects with molecular biology. It's connected with microscopy and so on. So for one person, solving a fundamental basic question is not very easy. So it's always recommended that you should form a team. You should collaborate with other. Like for example, someone is expertise in biochemistry. So for him or her, it's easy to solve the question of biochemistry. But now, how these biochemical changes give rise to a phenotype? This is so he or this phenotype, how to distinguish? So he or she need a help of a taxonomist. Then why this? Like, what is the molecular basis of such biochemical changes? So he needs the help of microbiologists. So this type of communication is very trivial to achieve good scientific achievements. So you must work independently. Also, you should be a team player. You should not think like, okay, this is mine. I will be the lead person. No, everyone is equal here. You need to treat them equally. You need to communicate them in a better way. And last not least, if you want to sustain in academia, okay, this is not very correct, but this is the fashion nowadays. If you want to sustain in science, if you want to be a scientist in academia, you must continue publish in paper. So publish means you should tell the scientific community, you should tell the globe that what you did. So why your work is, you think is noble. So you did something at your level, like at your institute, at your lab, but how can I know that what you did? So you need to publish it somewhere. So publish somewhere. There are some publishing platform which are world recognized already. So you should publish there, and that's how people will know it, that what you did, and you get recognition. And that's how in next level you go ahead. Why this is like this? Because to perform any kind of research, particularly life sciences, it costs a lot of money. Okay, so this money you need to organize. No one can give money from their pocket. No one will sell their land and give money to you. So there are particular organizations who fund you money. It could be government, it could be private, it could be some person. So when they will going to grant you money, when they see there is a clear perspective of what you propose that I will going to do. So you said, I will going to do something which will help the mankind, the society, or the individual, anything. So you need to convince them that this. So now you said, I want to do something in International Space Station, or I will do something on moon. Okay, the idea look like very nice. It look like something very fancy, but you must prove that you already did something similar kind of system, that they can trust you, that okay, if they will give you money, you can do something. So this is the point. You need publication, that you show that, look, I have these publications, so I think I can work on them. Okay, so okay, one thing I want to make clear. So if you have a doubt, you can stop me anytime and you can ask me question. Okay, so this is the fundamental scheme of roadmap in life science. So how we go further from one step to another step. So after doing bachelor's, so here this is bachelor's, you need to go for master's. Master's is MSc. So how you go there? You have an entrance test and some cases interview and so on. And also you need a basic qualifying grades in your bachelor's. So you are lucky, you qualify everything and you join for master's. Now, you join here in master's, you can do master's in any discipline of life science. You can do in microbiology, you can do in geology, you can do in botany, you can do in biochemistry, you can do biotechnology and so on. So after you do master's, either you can go for Master of Philosophy. Okay, I must say this MPhil things is very, very specific to either to India or to UK. This is not available in anywhere else. So you can go from MSc to MPhil. It's by either entrance, interview, and so on. Now, MPhil is entirely research degree, remember. Now, you can also do from MSc to direct PhD. You need to qualify certain exam. You need extraordinary research motivation. That's I said few minutes back, that you must know how to conduct research. So if you have a proper MSc thesis, it's always you have a better chance to join for PhD. You can convince the people that, look, I already did it, so you should take me. Also, after MPhil, you can join PhD. It's also by interview and your CV. CV means curriculum vitae. It's like what you did in your MPhil or your last studies. Like, for example, what the paper you publish, what techniques you learn, what topic you work, and so on. You can do from directly after bachelor, you can go for PhD. It's called integrated PhD program. There are some institutes who offer you this program. It's also relatively difficult. You need to qualify exam, interview, and so on. So after you finish your PhD, you can go for postdoc if you are interested in research career. So the postdoc, you also can do any discipline. For example, PhD you did, for example, in either botany or biochemistry, but postdoc people do in like a very particular subject. So someone do postdoc in, like, for example, certain kind of evolution in a particular organisms, or a chemical reaction, a particular chemical reaction, and so on. So here from PhD to postdoc, there are different ways. For example, you can write individual grant. So this is the point I said, that if you want to write individual grant, you need to convince the funding authority that you are eligible, you are potent to perform this research, and this is the research which you think will going to help the society in broad view. This is like, so always remember, you cannot say that I did PhD because I want to get a job. No one will going to take you for PhD. You should say, I want to do PhD because I am interested in PhD and I want to help myself to build my knowledge. And after that, I want to utilize that knowledge to development or fulfillment of the basic need of the society. The moment you said this, I want to do for job, your interest is not in PhD. Your interest in somewhere else. So be very clear with this thing. Then another way to join for research is you can apply for designated project. So what is designated project? Designated project means if I am a professor, I have a project to work on some certain kind of problem, like I want to work on artificial photosynthesis, assume. So I wrote a grant to funding agencies and they sanctioned me grant. They said, okay, we will give you this much money, so you can do it. In this grant, they give me, okay, they tell me, okay, you can take a student either for PhD, you can take for a PhD or postdoc, and this student will going to work with you for 4 years and this person will get salary and this person can work. So this is called like designated project. Now, if you want to apply for this type of position also, there are certain things you need to be considered. So one person, whoever it is, like a professor from India or abroad, they get hundreds of such applications. Okay. And they have position how many? One. So it's a very competitive. And it's not possible that you will going to go personally and talk to this person and convince. It's not possible. So what you need to do? You need to write the person such a way that he realizes that you are the correct candidate to join for that project. So you should tell what you did previous, how your previous knowledge will going to help to solve the question he asked in the present project. Okay. And secondly, how you fit with his team and his scientific community. If you're able to convince this, then he might going to give you a chance. Okay, let's have an interview or let's have a talk. Otherwise, you are in the beginning, you are straight away, you will going to be rejected. Number one. So for this is very important for your future I assume you are doing masters. So after masters, anywhere you want to do for academia, for scientific research, for PhD, or anything, anything, or job application. Even after masters, you can go for industry jobs. You should be very clever to write the letter, the cover letter. So when the email, a letter, a body you are writing, you should be very clever. Write less, don't write a lot. No one has time to read 10 pages. So write less, but very straightforward and very convincing. So this is very important. You just Google, okay, how to write a cover letter, how to write an application for a PhD, how to write an application for postdoc, I don't know. So you find something, you copy paste it, and you send it to 100 people. No one will go to reply you, because it's very clear. They understand that you did not read their profile. You must read their profile, what they are looking for. It's very important. And then design it such a way that they think that you fit with them. Otherwise, it's a very problematic, because otherwise it's a super competitive. So this is how it goes. But if you have a driven force that you are interested, you will do it. No one can stop you. So after masters, there are certain job opportunities. Like, for example, you can go to industries. Like there are certain industry, for example, if you did your masters in agrobiology, or if you do your masters in plant science, horticulture, and so on, you can go for industry like, like seed industry, like Ajit Seeds, for example, Myco, this type of industry you can join. Second, you can also go for government jobs. Like government jobs, there are recruitment in government labs like ICAR, Indian Council of Agricultural Research. They recruit scientists, but these are scientists B post, because you have a qualification of master degree. So, and also you can go for teaching jobs like school teachers. And there you have a job role, like you can be junior scientist, you can be entrepreneur, and so on. If you have a PhD, or if you have further research experience like postdoc and so on, you have more scope. But you are more scope, but it's much more, scopes are much more rigid. So for example, you can go for industry, you can go for government sector, you can go for teaching, like university teaching, you can go for research. Research means in different kind of lab, like CSIR lab, DBT lab, DST lab, like you can join NCL, NCCS, and so on. And there you will be like laboratory in charge, you will be assistant professor, senior scientist, and so on. But remember, if this is the perfect time that you should ask this question to yourself, whether you are really interested in research, because this is a very important question. Because after masters, if you do PhD, if you do postdoc, and then you said, okay, I think for me, it will be better if I will go to industry. It's not very easy. Why? Because you are overqualified and you need to get paid a lot. So they don't want to give this type of jobs in general. Okay, there are of course exceptions. If you are extraordinary, there is always exception, but it's not very highly, it's happening today. So your option for industry getting reduced when you do PhD and postdoc further. But after masters, you have much more chance to go to industry if you have experience of research. For example, like seed industry, if you have experience, if you did your research project from some company, some like some organization where you have in-hand experience how to handle these problems. So you have much more options to enter industry after masters rather than after PhD. So, but it's why PhD always I try to encourage, because this is the entry of research. Masters is not the entry of research. Masters, you have a theoretical knowledge of license. Like for example, the same. You want to drive car, you pass the theory test, like learner license. So this is a master's. So PhD means you have a practical test you pass that you manage, because PhD is, master is a time bound, right? that 2 years you have to finish your masters, but PhD is not time bound. It can be 3 years to 6 years, 7 years. It's not confined that okay in 2 years, because you have to finish your like objectives, what you propose, and you have to make some sense that how your work will be going to be helpful for the society. So you need to convince with the significance. So that's why it's a very like in-hand experience degree. So hope this is clear. So now, so types of job as a life science scientist, what you can get. So there are different types of job, depend on the specialization you have. You can be agronomist, as I said, that you crop production, soil management, and so on. You can be cytologist, that you can work on the cell function, cell structure. You can be ethologist, who work on behavior of animal and natural environment. Then you can be, of course, a botanist who work on plant science, epidemiologist, pattern and cause of diseases and how it's working, microbiologist, how the microbiome organisms are shaped, how it interact with other, and so on. Then geologist, you can work on solid, liquid, and gaseous matters and earth and other terrestrial planet, and then process, shape them, and how they interact with life forms. Marine biology, you can work on marine environment, like a sea environment, where what is the, the sea has a lot of organisms, like it's very diverse in terms of ecosystems and like ecological forms. So this is also a very good perspective. Paleontology, so the area where the scientists work on the fossil materials which are already extinct long back. Biochemist, this is the modern science, the structure, principle of living organisms, and so on. Because nowadays you like the definition of biology is changed. The definition of biology is now is the chemistry of biomolecules. So this is the very, very new science, I can say, no, old in new form, where it has much more wings. And the finally, like bioinformatician, you work on different computer programs like R, Java, the Python, MATLAB, and you try to incorporate different kind of data like transcriptomics data, say metabolomics data, phenotype data, so on, to build such a kind of sense that okay, this could be the way that it works. Like you develop a roadmap from this. So this is the job of bioinformatician. And I must say, today in advanced biology, like in globally, I can see it, the bioinformatician are the highest demand jobs, because everyone needs them. Without bioinformatician, advanced biology is not possible to execute. So this is something that if you get chance, please learn some like language, at least R, because without this, it's very difficult to sustain. So what are the skills you should develop to become a scientist? So you need your educational qualification. You should have a clear fundamental idea of your area. You should know how to do quality research. Your experience is very important. And you should always try to think something new outside the box. So these are the few skills and daily habit you must include in your life to improve your scientific caliber, scientific achievements capacity. So that I said, the documentation of your work is very, very important. So you should work on improving your writing skill. So this is another point I want to say here. Like in our system from India, so we suffer mostly, most of us, we suffer with the writing in English, because English is not is not our language, it's foreign language, right? So we never like learn it in that way. And secondly, which is I think is most important, that all of us, we have a fear on English, that maybe we are doing something mistake. This is something wrong we are doing. And because of that fear and shy, we never push ourselves. And if you want to sustain in science or any science, not only biological science, you must know how to write good English, because you need to write grant to get money. Without money, you will not able to conduct research. And after you finish your research, you need to publish it. So that's also again you need to write. And this is another issue. Second, you must have strong knowledge base. So the area you are working, you must have a knowledge of this area. So for example, and what you are writing, this is also important. You are working on plant biochemistry. Okay. You should not write something about COVID. This doesn't fit because you don't have knowledge in this field. You should write in your field. This is number one. If you have knowledge in your field in depth, then for you, it's easy to understand the lack, the part, what is missing, what is not known. And when you know what is not known, basically you solve 50% of the problem. So you write it down, okay, that I have, like this problem is not known yet. Then the third one is come, that how to solve this problem. So you try to think, okay, maybe with this way, you can solve. And then you find, okay, what techniques is needed or what way it's necessary to solve this problem. You are not familiar with this technique. So you should have an attitude to learn new things. Okay. So, and every time you learn new things, you should be more curious. Okay. So this is another thing. And always try to find a mentor. Mentor means who will going to give you supervise you each steps, like, okay, which direction to go. The mentor means he will not say you what to do. Okay. This is not the mentor job. He is not your boss. He is your like supervisor. He will guide you. Guide means you will tell this person, okay, I think this is the question and I have this 5 possible options to solve it. What is more convenience? And he can tell, okay, he thinks might be this direction. It's a discussion based. It's not a order based. So this is like how to become a successful researcher at every step of your career. Identify the right research project. What does it mean? You need to identify something which has an impact on socio-economical condition. So for example, now if someone will write like want to work on a research project on corona, like COVID, it will going to get grant very easily because people are really suffering with it. And if the person has a knowledge of the SARS virus and so on, it will be very easy for them to participate in these projects. So I think under this point, identification of research project, these are few key points. You should identify your skill. That means that I said what you are good with, what you like, what you are best, which subject, which topic, which technique, and so on. And resource, like from where you will get it. If you not know, from where you will get it. And recognize the requirement. So if you state a problem, to solve this problem, what you need, what instrument you need, what infrastructure you need, what facilities you need, what collaboration you need. Okay. Now, the research topic out of it, you need to build. And every moment you need to push yourself that how to get success, because you will get fail, but you need to boost that, improve this. Now, in this, you are working simultaneously on 4 research projects. You need to think now priority of them, which is more important now. I'm not saying that, okay, I will not going to work on others, but for now what I will work. Because remember, day is 24 hours, you need to divide it such a way. Because if you try to do everything, you might not able to finish nothing. So prioritize your time. What is now? and understand current scenario that I said, that the fact that what is more important, where you are standing, how many years left to submit the final report of the proposal, when you have a next meeting over this. So this current scenario and prioritize the time over this. And after you finish it, make a strong case. What is strong case? Means make a strong documentation of what you did. Like, this is the problem, this was the hypothesis, this is the research question I developed, and then these are the methods I took, this is the result I get, and this is the possible discussion, like, the interpretation of the data. This is also very important. Then, develop and nurture qualities of being successful researcher. So these are the 4 qualities of a successful researcher. You should have a courage to do it. You should be persistent. Persistent means I do it, I again do it, again do it. You should not give up. Then determination, reliance of face to challenge. You need to take challenges, okay? That you should not say, no, I cannot do it. No, you should say, I can do it, I will do it. And most important, self-motivation. As I already said, the scientific career path is long, tedious, okay? So most likely people around you not able to encourage you all the time. You need to encourage yourself. You need to motivate yourself that I will do it. I feel satisfaction when I do it like this way. And then again, you need to find mentor, because otherwise you will not able to simplify these things. You need someone who will, or someone or some book or something that who will going to give you suggestion how to take a right decision. You should understand your talent and try to enhance it. So you should always ask self-discovery questions. So what I am going to do in next 5 years? So, okay, I did my master's. So I will, next 5 years, I will complete my PhD. I will publish, try to publish this paper. And like this way. What is your strategy, strength and weakness? So in which fields you are strong and which fields you are weak. So for example, you are doing masters. So it's possible that you are strong in biochemistry, molecular biology, and the similar time you are not so strong, I will not say weakness, in, for example, breeding. So now if you know that your weakness is this, so give more time to this part and try to get some, like, more strength in this field. Which kind of project keep me excited? So for example, I'm a biochemist. So for my perspective, it's really fantasizing for me how plant synthesize glucose, because this reaction is not possible to perform in lab. So this is like kind of thing that you should ask yourself. Which kind of project excites you? Which strength would you like to cultivate in upcoming year? That's I said, like, okay, I am a biochemist, I'm a molecular biologist, I know about this technique. So next I want to learn something about spectroscopy techniques or microscopy technique or whatever. So this upcoming year I will going to learn. So this type of self-discovery question, like a roadmap, you must have in your head. Now, this is something important, like how to self-motivate yourself. Just imagine nothing will work. So what you will do? So that's why you need to self-motivate, because it's many times it's happened, and like I think everyone is very familiar with it. If you do experiment 100 different things, 80 to 90% time it will not work. This is how the life is. So this is not, you cannot put it anywhere. So this is just wasted. So you should not think negatively, you should think positively. So, okay, I failed. So, but I learned from this. Maybe next time I will do this, it will work. This will for sure, it will work, like this type of thing. If I solve this question, what it will contribute to the society? Like, for example, the global population now is 8 billion. So it's like 800 crore, 800,000 crore. So now this, like by 2050, it will be 12 billion. what they will going to eat? They will not have food. So if you will work something on sustainable agricultural development, on something on that like drought resistance, or you can do something that you will able to use the water resources, because we have 2/3 is water, so you can solve this type of problem. So which kind of journey I would like to go, like how I will go further, and how can I achieve this big win? Okay. So, and the most important, that how do I connect with people? How do I motivate others around me? And how do others will keep me motivated? So this is networking, like how you network your team. And research areas of interest. You should always enjoy working your field, strength your field, and how can you add new values to your institution and your research, a new thing what you are adding. So these are the parting wisdom. Be bold, creative, problem solver. Thank the people who made your journey. So you always think your previous teacher, previous mentors. Be satisfied with what you do. Understand your talent. Invest in there continuously. Begin with the end in mind. Your research is not money. I always said, research is not money. It's your interest. If you consider with the money, it will not work. Most importantly, don't forget to enjoy each stage of your journey. If you enjoy, then you can sustain. Otherwise, you will not be able to sustain. So this is the winning formula for this. Enjoy your work, take risks, network, don't overcommit, step away from science, and embrace future. What is the key question? These are the key qualities of a successful scientist. You should be passionate, you should be resilient, details oriented, but you're visionary, a creative thinker, determiner, knowledgeable, a team player, self-motivation, an effective communicator, and always think outside the box. This is the nutshell. All these characters are not possible to get together. Okay. So you should find your way. There is no formula such exist. You should find your way, how you can achieve there, how can you motivate, how you can push yourself. So these are the difference between scientist and researcher. So researcher cannot be a scientist, but a scientist can be a researcher while working, testing, verifying, and observing research project. A researcher uses scientific method in order to produce a solution, while a scientist conducts systematic organization investigation on a problem. A researcher can be any kind of field. It could be non-scientific, it could be literature, it could be education, social science, religion, etc. But a scientist must be from the field of science. Okay, clear? Now, here I would like to highlight the most 8 big achievements of biology that these techniques use in all field of biology, so you can understand how the fantasizing the life science is. So these techniques used in all of the life science fields. Many of these discoveries win Nobel Prize. Okay, and not once, more than once. Like restriction enzymes, you are read about in your masters or bachelors. Restriction enzymes. So they recognize specific nucleotide sequence to cut. So this is something, and this discovery restriction enzymes enable us to go for further cloning and all these things, to start to think on it. Then DNA sequencing. This is also another great discovery by like Sanger. Sanger's got Nobel Prize for this. So this is, here you can sequence the DNA and you can check their mutation, their changes, and what could be. And so it solves all kind of problem like genetic disorder and so on. Then PCR, polymerase chain reaction, discovered by Carey Mullis. So this enable us to amplify the DNA in faster way and organized way. So this, you are also might be familiar with this. Then gene targeting. So the targeting genes, it can be used for specificity, maturation, identification of different effectors, so on. GFP, green fluorescence protein. It was found from jellyfish, and this is a revolution in microscopy. Like for example, confocal microscopy, light microscopy. If you want to see where my protein is localizing, where this protein is interacting with other. So these are the tag. Like this is the protein tags. So it enable you to see your protein under the microscope because it's emit green fluorescence. So later on, there are a lot of mutation they did, targeted mutation to change the green fluorescence to red fluorescence, become RFP, to yellow fluorescence, become YFP, and so on. The next RNAi, it's RNA interference technique. It's allow us to knock down gene of interest expression and to characterize the genes. Then iPS cell is induced pluripotent stem cell. So you might heard about the stem cell research. So this is something that it can develop from somatic cell, and it has a potentiality to differentiate it into an organ. And as it is a pluripotent stem cell, so the body accept them, not refuse them. So that is the magic of it. Otherwise, other normal somatic cell body will not accept; they will reject it. And finally is the CRISPR-Cas. So CRISPR-Cas is a very recent method. It's like 10 years. It's allow you to target and specifically edit the genome. That's why it's called CRISPR-Cas mediated genome editing. Because you might know that transgenics are not used by the society because of lot of issues. So because of transgene and so on. But CRISPR-Cas is not a transgene. It's editing the gene such a way, either gain of function or loss of function. Gain of function means your one gene is giving you a product of what you want. So if you induce this gene activity to modifying the promoter or so on, you can enhance the production. While if certain gene give you a detrimental effect, like because of this gene, your product is susceptible. So you can mutate that gene, so product will not be any more susceptible. And this is the discovery in 2020, Emmanuel Carpentier from Max Planck Institute in Berlin and Jennifer Doudna from UC Berkeley, they got Nobel Prize in chemistry for this. So you understand how these techniques are like important in our scientific community field and so on. So, okay, this is the last question here. How much do biology research scientists earn? So, biology science, like this is about salary wise. First of all, you cannot compare with the salary of academics with other, because, okay, here I want to say, you have education, that doesn't mean you will earn more. It's not always the education is directly proportional to the money you earn. Basically, there is no direct correlation. There is no correlation at all. So in abroad, in developed nations, the salary for biological science researchers is about 3,000. Yes, it's about 3,000 rupees per hour. While in India, for example, the salary depend on different jobs, like if you are in junior scientist or you are assistant professor and so on. So this is the kind of the salary scale, but it can be increased also, and depend on your experience, depend on your quality, the need of the system and so on. So this is the fundamental, like overall, that how much you can earn. Okay, thank you for listening. You can, if you have a question, you can write me. This is my email if you have a question.
Okay, thank you, Dr. Saintan. You have almost covered all the things which I had been told to you to cover up. So if students had any questions, then they can ask, or as Dr. Saintan said, they can email.
No, They can ask me now also without no problem.
yeah.
They can ask me, and okay, one thing, they can ask me in any language. They can ask me in Hindi, they can ask me in Marathi. It's not a problem.
Yeah, the students, those who have the queries, you can put your queries in the chat box. Okay, I think as of now, we don't have any queries. Iris, sir, should we move further?
Yes, sir. Yes, sir.
Yeah.
I think Sayantan, I have. Yeah, Parshuram here. Hi, Sayantan.
Hi.
It was nice talk, Sayantan.
Thank
Yeah,
you.
I think so. You covered almost all the things. I would like to add one point into it. The English. I think this is very important thing which the people, the college students generally always will be skeptic of. I think the college environment also should be a little bit strict about it, because what happens that we always try to get in our zone of comfort, and then when we do present, or the student present the work, or they communicate with the faculty, they most of the time they start speaking in the Marathi or regional language. I think it's a good point if they can focus on that this time itself to practice or the communicate more in the English in these stages itself. I think that can help a student a lot. I think that's what I can add about it.
I think, Parshuram, you are very correct. But what I want to add it with this, actually, your language skill will not improve till you don't get a force. You understand?
Yes. Until you come out of your zone of comfort, actually.
Like if you don't have any other choice. For example,
Yes.
I can tell you my experience.
Yeah.
I am a guy from Bengal. I came to Pune to do my masters, but I can speak Marathi. I can listen, like understand Marathi and everything without any problem. Because in Pune, without Marathi is very difficult to survive.
Correct.
So this is the force you need, like that. Okay,
Yes.
you need to do it.
Yeah, same thing happened with me when I went to IIT Madras first time. So there was no other option. Like even people were not even speaking Hindi. So that was the thing. And then within few days, I think we started understanding. But it's like even though you want to learn, you need to put efforts. So if you say that, like the first thing I did in Chennai was to buy the grammar book and learn that by heart. The 15 days were essential. I spent time with that, and within 15 days started practicing with the people. And I think that's what I said also, like student needs to do this in the college level itself.
Like I want to tell, as you open this point, actually we limit ourselves. Like yes. I can see in the students, because it's everywhere. People are always try to blame others for something, not themselves.
Yeah.
Okay. Like, okay, I did not get good teacher. I did in college. We don't have this facility. We don't have this.
Yes.
Here, everyone came, particularly in academia. I see from India, particularly the people in academia came from lower middle class background.
Yes.
Everyone.
Mostly rural background.
Rural or lower middle class, because
yes.
this 15 years journey, if someone have money, they will not go for here. Why they will come?
Correct.
We don't have option. That's why we came. So here you need to think that you need to push yourself.
Correct. So yeah, I think this is very important. And even I think, as I remember, Shekhar Mande was telling once that most of the scientists in even India, so at least 60 to 65% of the population of the research scientists comes from either middle class families, small towns, rural areas, or like very remote areas, sometimes you have never heard of.
Yeah, so I came from such areas.
Yes, we are also from the same region. I think Mahendra also knows that. Like many people even in Pune, I think we had tough time to explain that we are part of Maharashtra also. So I think this happens. So I think, but people needs to talk about this, and I think talking is the only way practicing is only way. I think that's
like this is this,
research.
this,
I
this
think this is one more point you covered in the research. I think that is very important, where frustrations are part of researcher's life, and you need to really motivate yourself with that. So yeah, when you do things, and I think people are very, this new generation is very impatient. Even their mobile doesn't work for long, they get really impatient with that. And then if you see that, research is different thing. Your experiment, I remember I had failure for continuously 1.5 years, and it was tough time to motivate ourselves. So I think this is very important, and that is very important point. If you really want to get into research, prepare mentally.
Actually, like this is the difficulty. You said, like, in this type of forum, when we talk, we expect the students should say, tell something. I am not a tiger. I will not going to eat them. Come on, tell that what is your problem.
Yes.
And I am damn sure they have a lot of problems, but they will not going to say it.
Yes.
How, if you not say it, how can I solve it? Right?
Yes.
So this, and second thing, which I think is very important to understand, what you said about patience and like in research, if you look on the research career perspective,
Yeah.
from our childhood, we get an education that you do academia to get a job. Okay. So a PhD, when all of us were doing PhD, whoever, like, I think it's for everyone, like for you, for me, for Mahendra and everyone, your family people will ask you, so when you are going to finish? What it is? How can I say when I will going to finish? You cannot say it, right? It's very difficult.
Those are like really different domains actually. So apart from the basic research what we face, I think this is another level of problem.
No, because that's why it's PhD, no? That's why it's PhD, Doctor of Philosophy. It's a philosophical degree. You need to reach, like, that level.
It's good.
Dr. Sayantan, there is a question that, can you share your research journey? So can you put some light on your own journey of research?
Okay, so I can say I don't have a slide, okay?
No,
So
just in within 2 minutes, we can have a
slide
journey, how you started and ended up with that.
So as I just said, that I came from a very rural place of West Bengal. Like, my place still doesn't, it's not have any road. When I did my schooling, there was no electricity even. So this is the condition. And from there, I came, like, I did my bachelor's from there, from West Bengal, and I came to Pune. So when I came to Pune for my master's, so I came for the entrance exam, and that also I came after fighting with my father, because my father was not willing me to send outside. This was the condition. So I did my master's from Pune, and after finishing my master's there, okay, so I did, during my master's, I did summer training from DRL, Tejpur, Defense Research Laboratory, Tejpur. And I work on biodiesel production. Then I did my master's thesis on agrobacterium mediated transformation in Artemisia annua. Artemisia annua is a plant produced artemisinin, which is anti-malarial drug, and I think 2019 they get Nobel Prize for this drug production, because nowadays quinine is not working. So this is my master's, and then after master's I joined for master, like MPhil. Also with MPhil, I was simultaneously working on a UGC project about host pathogen interaction. So for my, so this point of my life, I work on 2 independent projects separately. So although, like, I was working at a time. So my MPhil was on like population genetics of a plant, a weed like a plumbago. So I was traveling all across the India, from Kashmir to Kanyakumari, from Arunachal to Gujarat, to getting these plants. And these are, these are weeds. So they are not from nursery that you go and someone will going to give you with a bag. So I was hunting them from the wild, from the forest, and I was putting them in the soil, bringing them back to the like my institute, like Pune University. I was growing them there. So Then I work on this for almost 3 years. I managed to publish something from this. And also then after finishing this, I joined for PhD immediately, and I continue with the PhD with host pathogen interaction, which I was working as a UGC project fellow. So my PhD was on tripartite interaction, like tripartite. So I need to tell. So bipartite means interaction between 2 organisms. Tripartite means interaction between 3 organisms. So I did my PhD with a tripartite interaction, where one was like a host plant, one is a pathogenic fungus, like which cause disease, and another one is endophytic beneficial fungi. So this is another one is also fungus, but it benefits the plant. So this 3 component, how they interact, how the molecular changes in the host, how it works, how the genetic remapping, and all these things. And from there, when I was working, I published some stuff, and I identify the role of certain phytohormones signaling in this kind of tripartite interaction. Later on, when I joined Weizmann Institute of Science, I continue in this hormone signaling pathway and how it works on the production of toxic metabolites. And so here I am working from last 7 years in this institute where I am working now. So I publish things. So my primary interest is about how different like omics networks, like, you know, omics means transcriptomics, like genes, metabolomics is like small molecules, hormonomics like hormones, and so on. So I work on transcriptional and post-transcriptional. So a gene expression and after the gene expression regulation. So this network model with their secondary metabolite production, how it governs to a plant fitness in a changing environment. Changing environment means when it interacts with others, like it's either it interact with environmental stress or it interact with biological or stresses. So here, this is the topic I work. I also work on the molecular cues and hormonal regulation of ripening processes, how the ripening govern and what is the regulators of the ripening. And also I work on the pigment biology, like the colors. Like, for example, you know, I work like if you go and buy eggplant, like brinjal, it's a purple color. So how this purple color originated? What is the consequences? What is the molecular regulation of this purple? How this purple color is accumulated? How it's degraded? What is the biosynthesis regulation, and so on? So this is a kind of the field I work with. Okay.
Okay, thank you, sir. Thank you very much.
Thank you.
Now I request Dr. Pankaj Mundra to propose the vote of thanks for both the sessions.
Yeah. Thank you very much, sir. So at the end, I would like to extend a sincere vote of thanks to both our resource persons, Dr. Parshuram Sonavne and Dr. Sayantan Panda, for their valuable guidance. Talking about Parshuram Sonavne sir's lecture, it was really a wonderful lecture. It was a treat to listen to you, that what kind of research should be done, what depth one should achieve in the research, and what are all the things one should keep in mind when doing the research. Also, regarding Sayantan Panda sir's lecture, it was more into research philosophy and psychology. So it was really a important aspect that one should understand while doing the research. The patience is the most important thing that students should have. And especially during this pandemic situation, both the resource persons accepted our invitation and gave us their valuable time and guided our students. So I thank them on behalf of our institute. I would also like to thank our principal for allowing us to conduct this webinar, and Dr. Mahindra Ahire sir especially for arranging those very nice 2 resource person. I would also like to thank all the participants, and with the chair, I declare that this session is over. Thank you.
Thank you. Thank you very much, Parshuram, Sayantan.
Thank you, Mahendra. Thank you.
Okay,
Thank you, everyone.
okay. So now we can leave the meeting.
Yeah.
Yeah.
रजिस्ट्रेशन पण झालं असतं पण ते मध्ये प्रॉब्लेम आला त्यांच्याकडून आता कडनं काय नाही मला माझं सगळं ओपन होणार आहे ना ऍप्लिकेशन ओपन होतं पण लेट पण ओपन नाही ओबीसी काय मला मिळत ते नावात माळी बघून काय तर मी बाय डिफॉल्ट कन्सिडर केलं ओबीसी ते नंतर बाकी त्यावेळेस